This is a special episode of the Immunology podcast, Immunology 2025, Day 5. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Rao. Welcome back to the Immunology podcast, where we have conversations with immunologists. Today we're back with our final episode from Immunology 2025 in Honolulu, Hawaii. We'll be discussing highlights and sharing our thoughts on how it all went. If you're tuning in for the first time, be sure to visit immunologypodcast.com or your favorite podcast app to check out our episodes covering the first 4 days of the meeting. I'm going to kick off things in just a minute, but before we get to that,
whether you're looking to attend an immunology conference this year or to expand your network, make the most of your experience by downloading our collection of tools to help you prepare for your next event. STEMCELL Technologies' downloadable checklist and guides include recommendations on how to get ready before attending conferences, tips for networking, best practices for your LinkedIn profile, and more. Download the conference toolkit at www.stemcell.com/conference-toolkit. Last day, Jason.
it is. We just finished the conference and are doing our last recording here.
I know.
And I'm going to the beach this afternoon.
Yeah, sounds like a good plan. I'll probably follow that.
All right. Well, I got a couple talks from this morning.
Mm-hmm.
I kind of bounced around.
Mm-hmm.
The first one was Immune System Crosstalk. It's really just a shout out I'm giving for this talk. It was called Engineered Tissue Tregs Emulate Inflammatory and Ischemic Diseases by Payam Zarin from Genti Bio. They talk about how Tregs can lose function due to lack of growth factors, and they're doing engineering, which they really quickly gloss over, unsurprisingly. To overcome that, they have a dual editing approach, and they have some strong mouse data for type 1 diabetes, IBD, and ischemic stroke, and they're getting ready to go into people soon. I tried to capture how they were doing all the editing, and I'm pretty sure there was not enough detail for me to capture how they did all the editing.
It's a trade secret.
Then I went to a major symposium on dendritic cells and have a new favorite talk, which is Ann Anderson from Harvard on some crazy coolness going on with opioid receptor, which is one of my favorite things in the universe. So what they found was, they got to this really interestingly. They used Slingshot, which is software, to look from naive cells all the way through to cells that stop functioning, exhausted cells with high levels of PD-1 in tumor progression in a melanoma model. And then they noticed that there was this, they looked at gene changes over time, and out of this came proenkephalin, which is endogenous opioids. It induces CD8 dysfunction, is what they found. And its target, and they looked at targets, because this is the endogenous, one of the endogenous opioids. It can target all the opioid receptors, delta, kappa, and the canonical mu. But they weren't highly expressed in the cells they're looking at. And they're like, well, that could be fine, because, single cell RNA-seq and RNA-seq in general doesn't pick up low-level basal transcripts. But they took a little further, and they found also that there was this opioid growth factor receptor, often known as the zeta opioid receptor. And that was the key here. And they found that what was happening was and then they showed that gain of function lead to failed T cell control of tumor growth and poor effector function, and loss of function, you had better killing of the melanoma. So they got some functional stuff and looked at, and they just floxed OGFR now. They can do the regular knockout, but they have a floxed one now. But they're able to show that if you do peripheral antagonism of opioids, general opiates with Xelopran, which was being designed for opioid-induced constipation, plus PD-1 blockade, you have enhancement of activity. And what they're really getting at is that, and then if you look at clinically, acute active opioid signaling correlates with poor outcomes. So they're seeing that opioids are driving an immunosuppressive state in T cells, leading to worse cancer. And this ties into other work that's been going on, kind of known and keeps getting forgotten about how wound healing is inhibited by chronic opioid use. And chronic opioid use is immunosuppressive, but acute opioid use is often opposite because the receptor class switches depending on acute or chronic signaling. And so it got really interesting, and I ended up talking to them afterwards because I nerded out a little bit, I'm not going to lie. But it was really interesting, and just like a common drug class, opioids, right, used in cancer all the time for pain, could be making prognosis worse, which is what we see in other diseases.
Oh man, that's terrible. You're taking opioids to help you cope with your cancer, and then might be
killing you.
Yeah.
Yep.
Oh.
Mm-hmm. And then one more, Zachary Hay from OHSU, Oregon Health Science University, was looking at androgen receptor-mediated suppression of CD4 T cell anti-tumor immunity. So super high level, we know females tend to have more autoimmune disease, and males tend to have more disease from a failure of the immune system to respond. And he was looking at, well, that's a sex difference. What's going on? Is that an androgen receptor thing? And it is. And he found that androgen deprivation reduces Treg expression of NRP1 in the tumor microenvironment, and that it relates to chromatin accessibility when androgen receptor is expressed and ligand is present. And they're trying to figure out how that then regulates Tregs because they see the dysfunction. So, if you have a lot of androgen there, you are going to have, get pulling off androgen. So being more female leads to less Tregs and more immunity, whereas in males, you're suppressing Treg. So, right, because males don't kill and women overactivate their immune system is the kind of cartoon of this. Yeah. So they're looking at the male receptor here and trying to see how that was causing a difference in sexes. So that was really cool. So more immune crosstalk for me today. But opioids.
Very nice. Is that it?
Mm-hmm. I'm done.
You're done? Okay, very good.
Take a fork at me.
So I want to talk a bit about technically yesterday's talk. So because we recorded before the presidential program, the President's Symposium, sorry. So I just want to just briefly talk about a bit of what was shown there. So this is so the president, Stephen Jameson, he selected a few people to give their talks, and I thought it was pretty cool. So first was Marion Pepper from the University of Washington School of Medicine, who studied allergic asthma, and basically she uses this mouse model of antigen, asthma against house mite antigen called ERP1. And what's interesting, like the idea that she wanted to address, is that we know that in asthma, patients with asthma, the asthma really kind of seems to live in the lung. And she mentioned that patients that receive lung transplants, then if they're asthmatic and they get a new lung, they don't get asthma. So they don't immediately develop asthma. So it's really, there seems to be tissue resident cells in that lung that are important in this kind of chronic asthma. So she was looking into so Th2 cells in the lung, and basically these cells that are expressing the IL-33 receptor, ST2, really seem to be the ones that are making kind of these pathology related cytokines. They are a heterogeneous bunch. And so they site-seek to look into the heterogeneity, and they find that there's this population of TCF7 positive cells that are that don't express. So there's a differentiation. The TCF7 seems to really mark two different, in a broad description, two different populations. And the cells that don't express TCF7, they do express cytokines that are related to the pathogenesis, and they reside outside of tertiary lymphoid structures that you can see in the lungs of asthmatic patients. This other population of TCF1 or TCF7 positive cells, so TCF1 is this protein and TCF7 is a gene, they are more likely to be in the tertiary infrastructures and to require kind of the B cells to support their development. And these cells don't express so many cytokines, but are probably the precursors of a more terminally differentiated BLIMP1 positive, IL-33 positive Th2 effector cells. So you have like this separation again of an effector population, and then a more long-lived population that probably feeds into the effector population, and these are residing in the lung. Dana Farber from Columbia University was next, also talking about tissue resident memory and the importance about the timing of the generation of tissue resident population against pathogens, and this idea that you have sites. She compared tissue resident memory cells around different organs. And there's like specificities about these resident memory cells living in the gut or in the lung. And basically there's these three phenotypes: gut, lung, and lymphoid tissues that seem to be pretty distinct groups. And she just talked a bit about the idea of having your first kind of infection and your first wave of tissue resident cells, and that these are different from what happens after a new infection. There's these newcomers, as she calls them, seem to occupy a different niche compared to the original cells, the OG cells that responded to your first encounter with this pathogen. And there seems to be some kind of competition, and that seems to kind of the cells find themselves in slightly different locations in the tissue, and in a slightly different interaction with like the vessels and the sources of the communication of the tissue with the general circulation. So basically, the first wave and second wave are qualitatively different. Also, as John Hardy from Iowa University talking about T cells in the brain, and the idea that we always thought that we don't have much immune cells in the brain. This whole immune privilege concept, and I think that we have really moved away from that. But mouse models are still a bit difficult, and she showed that specific pathogen-free mice that are the way that we usually have our mice, host our mice in the mouse facilities, actually those guys don't have a lot of CD4 and CD8 cells. And a good way of understanding the infiltration of particularly T cells in the brain is having mouse models that are, quote unquote, dirty, that they have a bunch of other microbe colonization. And he shows two different ways of making these mice, either co-housing them with pet store mice, or like mice that have a very diverse potentially pathogenic microbiota and a lot of diversity. But he also did the sequential infection, and he infected, I think it was mostly viruses. I don't remember seeing any other any bacterial microorganisms in that list. But it's mostly sequential infection with viruses, and that this really induced the accumulation of T cells in the brain, and he showed that many of these are specific to the pathogens that he introduced. He could do that in this when he did this sequential infection, so he knew what he was infecting with. And then it is really help look at where the cells are going, how the T cells look, and the kind of markers that they are exhibiting, and how they are interacting with microglia and resident dendritic cells. So basically, these mice that had T cells in the brain, in a situation of inflammation, you do see some disadvantage in terms of like seizures or some affecting kind of the brain function. And mostly because of interferon gamma production by these cells in the brain seems to be something detrimental. I think there's also a lot of other research he didn't mention, but from the papers that we've been covering, that makes sense. Interferon gamma in your brain is bad, like really bad. And I think that's just really nice how it interacts with oligodendrocytes and insulation of the neurons, and how it kind of interacts with this brain structures. But you can only really see this if you have mice that have a much more diverse and slightly pathogenic microbiome. And I just also want to say, so at the end of the talk, we also had a special talk by David Schatz from Yale, and I thought it was really cool because he talked about the role of recombination in the production of immunoglobulins, and I guess also T cell receptors, and how we came about to understand how this whole thing works. He mentioned that it was 1976 that Japanese researchers found that you have the IgG genes, or the immunoglobulin genes in B cells, are recombinated, so they're not the same as the germline. And that was a big mind-blowing discovery. People were not expecting that. And so obviously the question is, well, how do you recombine these genes into this final production and this final presentation of the gene? So so David was involved in the searching for this recombination machinery. He worked at David Baltimore's lab in the 80s, and he actually kind of found these VDJ recombinase, and using a very kind of clever proxy for analyzing recombination activity. Basically, he had this. It was really weird. He put this reporter of recombination. He designed a really nice reporter that could show if a cell had been subjected to recombinase activity in fibroblasts. And then he basically transfected a bunch of fibroblasts with like pieces of genomic DNA until some of those fibroblasts showed, this reporter for recombination. And then they had to do a genome walk. I'm thinking that this feels really hard, really old school, a genome walk. And And that's how they ended up identifying this RAG1, RAG2 locus, which is really close to each other, which is very compact. So it's a little bit unusual for a eukaryotic gene. And basically, they show that indeed this was a this RAG1, RAG2 locus is the leftover from a domesticated uh, um, transposase element that he showed through evolution how they think this thing ended up uh falling into this new function for IgG, or for recombination of immune cells. So I'm not going to go to details, but he showed such an interesting kind of story of how we understand this rag genes and how they work and their structure and all the structure analysis of how actually the DNA has been cut, and what is the evolutionary kind of red line that brings this enzyme to the current state, and how analogs look in other parts of the animal kingdom. So very, very interesting talk. And I just want to go very quickly through like the morning talks today. So I attended the session on lymphatic immunophysiology. And yeah, I don't want to be too long, but basically it was really cool. So talk with a lot of like imaging about the lymph, and a lot of talk about the lymphatic vessels and all the things that they are involved with, and how important they are, and how is our understanding. It's actually not so good how we understand the role of it. So we had Sandro Damesquita from Mayo Clinics, who was talking about the importance in brain physiology of the meningeal lymphatic system, and how important are those lymphatic vessels, and how draining antigens from the CNS, and how they relate to aging, and that you have a dysfunctional drainage that is correlated with aging, and this actually can be important in impaired cognition, and how it impacts things like lipid composition in the brain, and basically how important lymphatic drainage is for brain function. There was also Gwendolyn Randall from the University of Washington School of Medicine in St. Louis. So she was also talking about the role of the intestinal lymphatics and in gut immunophysiology, and how important it is, for example, for draining things like HDL. They go through the lymphatics, and she talked about what we understand of that, of those, or that relationship. Ian Fleming from Colorado talked about, also stromal cells in the lymphatic endothelial cells, and how important they are for storing antigen from a vaccination. How can they really be a good cell to target for like long-term antigen procurement after vaccination, how to kind of mediate a long-lasting immune response against this antigen. And then basically also the role of cell migration through non-lymphoid tissues. So this was not so much about the lymphatic vessels, but about the kind of things that we understand about T cells and how they move between lymphatic, non-lymphatic tissues, and the role of different molecules in this. And then I think the last one I want to say was really cool was from Eric Song from Yale. And he had this really cool story about the role of lymphatic drainage in the eye, and how important it is to have proper lymphatic drainage, and how difficult it is to if you want to stimulate that. She showed that stimulating lymphatic drainage can help with edema in the eye and help with glaucoma, and a lot of these conditions of the eye can be helped. But then it's really hard to induce lymph angiogenesis, all right, without also inducing angiogenesis. And angiogenesis, so it would just like that's just blood vessel growth. That's not good. That doesn't help. So they developed this a mutated version of VEGF, which induces angiogenesis. It uses lymphoangiogenesis, VEGF-C, and they usually induces both angio and lymphoangiogenesis. So they made a new version of it that is very specific at inducing lymphoangiogenesis, and this can help reducing swelling and lymphedema in the eye, but also in other settings. So I thought it was really good, the talks this morning as well. But man, I'm ready to take a little holiday now.
Coffee time.
Yeah, coffee time for sure.
All right. Well, that brings us to the end of our Immunology 2025 episode series. We've met a lot of people and had a lot of fun, and can't wait until 2026 where we see you all in Boston. Don't forget to subscribe to our newsletter at www.immunologypodcast.com. You can also reach out to us X on @immunopodcast, Blue Sky at immunologypodcast.com, or via email info@immunologypodcast.com with feedback. See you next time.